changes. These changes make logical sense. For example, when I get to the end of the respiratory pathway, in the alveoli, where gas exchange is going to take place, what kind of epithelium will I find there? When I get to the end of it, all the way out to the end of the respiratory system, what kind of epithelium is there in the alveoli? Okay, are y'all following me? This is anatomy content, so you're learning this stuff, right? I've got simple squamous there. Those are single layers of flat cells. Now the capillaries that are surrounding those alveoli, what does their anatomy look like? Simple squamous. So if I'm going to move oxygen and carbon dioxide from inside the alveolus to inside the blood, how far does the gas have to go? It has to cross two simple squamous epithelial cells. Now, why wouldn't you want those to be cuboidal or columnar? Well, because I don't want these guys to make anything. You follow me? I want them to be as thin a barrier as I can possibly create. It's as though the two compartments are almost continuous with each other. And since the gases are lipophilic, they move across the membranes easily, it makes gas exchange incredibly efficient. The anatomy speaks to the function. It makes sense. The anatomy makes sense. Now, why would it make sense then for the small bronchi, so I'm down there close to the alveoli. Why would it make sense for them to be ciliated? Okay, well, you don't know this yet. I'm going to talk about it more on this picture, so I'm going to tell you this. It makes sense because these columnar and pseudostratified columnar layers tend to have cells in them, these especially, tend to have cells in them called goblet cells, which make a mucus-like product. So if I were to put a mucus-like product on the insides of cells, whether they're columnar or pseudostratified columnar, and then I put cilia on them, the cilia, you already know, have the ability to move, right? They were built that way, out of these proteins, and they can move. What would you want to be doing with the mucus that's created down there near the bronchi? So I'll put these column cells on there so I can stick cilia off of them, and they're moving. They create a river, if you will, a coating, a mucus coating on the inside of those small bronchi, and those cilia beat in such a way so as to move product up the respiratory tract toward the larynx. The cilia beat in a coordinated way in order to do that. So as we breathe and material moves into our respiratory system, this serves as a first line of defense for the immune system. It makes sense for these things to have cilia on them to move that product out of the respiratory system into the digestive system. That is their ability. All right, so that makes sense for the small bronchi. What about the uterine tubes? So the last section of this class, I'll show you these pictures of the uterine tubes. The ovaries in the female reproductive system are not connected to the female duct system. The fallopian tubes and the uterus do not have a connection to the production of the sex cells, the egg cells. Instead, there are these finger-like structures that drape over the ovaries, called fimbriae, anatomy meaning fingers. And the inside linings of those fimbriae are simple columnar cells that are ciliated. So as the egg is ovulated each month into the woman's pelvic body cavity, there has to be something to draw that egg into the fallopian tube. Otherwise, there will be no more humans. How does that happen? You see the importance of cilia? They create currents to draw that fluid out of the pelvic body cavity into the woman's reproductive system. into the tubes. All right, and similar function here in some regions of the uterus. Ciliated and non-ciliated columnar, simple columnar cells. All right, here's the next one. We've talked about this one a couple times, so now here it is. There are two varieties here also of pseudostratified columnar. We've talked about the ciliated. The non-ciliated types are in the male sperm-carrying ducts. Now, when I always introduce it this way, so I introduce the fimbriae of the fallopian tubes and the ciliated columnar cells there to students. And on the next picture, I say the lining of the male reproductive sperm-carrying ducts is non-ciliated pseudostratified columnar. And students look at that and they go, don't the sperm cells have to move? Don't they have to move through the system? Well, certainly, they most certainly do. So what causes the egg to be drawn to the fallopian tube for reproduction? Ciliated, simple columnar epithelial cells of the fimbriae. What causes the sperm to move through the male reproductive duct system? Well, let's see if we can put it together. I haven't taught you this yet, but I told you blood vessels are lined with epithelium. I told you the next layer in the blood vessel is smooth muscle. And as it turns out, all these ducts, and we're going to find this over and over again, respiratory, digestive, reproductive, urinary, see it over and over again in these tubes. The walls of the tubes contain smooth muscle. And the smooth muscle is arranged normally in sheets connected by tight junctions. One sheet that goes around the tube this way, and one sheet that goes around the tube this way. Now I'm going to say this again when we get there, but I want to alleviate confusion now. A circular and a longitudinal layer. And those layers will contract in an alternating fashion, longitudinal and circular, in a peristaltic type fashion in order to move the contents of the tube forward. And we'll learn that first in the digestive system. But it is also found in the ducts of the male reproductive system, the alternating contractions of smooth muscle within those ducts. Okay, and then the ciliated variety we've talked about. These guys line the trachea and most of the upper respiratory tract. Okay, have you learned now some of the transitions in the respiratory system? If I told you I've made a cut in the respiratory system and I put it on a slide, what kind of lining tissue would you expect to see there? This kind. Most of the respiratory system looks like this. Pseudostratified, ciliated columnar cells. What if I told you I cut through the alveoli? What would I find there? Simple squamous. What if I told you I cut through the small bronchi? What would I find there? Ciliated, simple columnar. Okay, now there's one more piece, and then I'm going to do that one more time with you, the transitions in the respiratory system, one more piece to that, and it's this one. Okay, so we've moved from the simple into the stratified, and so this is an example of a stratified squamous epithelial lining, and look at the example that I use here. This is the esophagus. So if I had a picture, if I had a histo picture like this, and I had the picture at the junction of the esophagus and the stomach. And so the picture over here continued as stomach lining over here. And I put an arrow right here and I asked you to identify this junction. How would you know that this is esophagus and this is stomach? It is a major anatomical shift in lining from the esophagus to the stomach. And I've said it 3 times today already. What happens? We move from stratified squamous into what's lining the stomach? Simple columnar. So this really thick layer here of esophagus becomes like this. You get a dramatic shift in the lining from stratified squamous into simple columnar, from the esophagus into the stomach. Now, stratified squamous actually exist in two varieties, not ciliated and not ciliated and non-ciliated, but in this type in the presence or absence of the protein keratin, which is a fibrous protein. Some of the tissues in our body that are stratified squamous are non-keratinized. These are the tissues found inside the esophagus, inside the mouth, inside the nasal cavity. Add that one here so that we can do the whole respiratory system. In the vagina, in the female reproductive system. Non-keratinized stratified squamous epithelial cells inside us. Now, if I tell you that the nose is lined with stratified squamous, non-keratinized stratified squamous, at this point we should be able to put together the entire respiratory system. So let's get on board Miss Frizzle's school bus and make our way through. Yeah? As I pass through the nose, I point out to the students, look at the lining inside the nose. What does it look like? And all the kids say, non-keratinized, stratified squamous epithelium. And I move from the nose then into the pharynx, into the nasopharynx, and down into the oropharynx. And I make a turn there, and I pass through the voice box, through the larynx into the trachea. And as I'm passing through these areas, through the pharynx and through the larynx and into the trachea, I say, students, look outside. The lining tissue has changed. What is it? Ciliated, pseudostratified columnar. Now, as I pass from the trachea into the bronchi and I get closer to the small bronchi within the system, I say, students, look, it's changed again. They're not pseudostratified anymore. The layered appearance has disappeared. What does it look like now? It's simple columnar. Is it ciliated? Yes, it is. And as I pass from that layer into the little places where gas exchange is going to take place, I say, look, it's changed one more time. What is it now? Simple squamous. The transitions in the epithelium and the respiratory system. A great example of all these types you're learning. Now, the keratinized variety is the epidermis of the skin. And so in the next chapter, this is where I will begin in talking about skin, the anatomy of the epidermis. The skin is made up of two layers with an underlying adipose. The layers are epidermis and dermis. And the epidermis is this, stratified squamous epithelium. Now, this is beautiful here. Students have no trouble imagining this is all flat cells. But I'll point out to you something in the skin that's going to bother you a little bit. Bothers me a little bit. We say the skin is by definition epidermis stratified squamous, but the basal layer and the spinosum does not look squamous to me. We're going to have to go with the definitions, things in anatomy that bother us, right? So it's stratified squamous. It's keratinized, has a high content of keratin, and I'll mention just very briefly some language there to help you remember the transitions in keratin. Essentially, the last 20 or 30 layers of the skin that we call the corneum is nothing more than dead cells filled with this protein, ready to be sloughed off. Okay, so non-keratinized and keratinized. Now, at this point, students want to know, this is what you want to know, why didn't you talk about stratified cuboidal and stratified columnar? And the answer is, they're very rare. I'm not going to waste any time with them. So we're moving now toward the end of our epithelial discussion. The next picture is connective tissue. So how do we finish this? Well, I think it's appropriate. Every good textbook finishes the epithelium with transitional. This is because you think at this point that you can explain everything. If I look at an epithelial tissue, I can explain it. It's either flat, or it's cubed, or it's column. It's one layer, it's multiple layers. It's ciliated or it's non-ciliated. I can describe the epithelial lining. Okay? But there are some places, for example, in the ureters and the bladder and the urethra, in the urinary system, where it's not so easy to describe what it is you're looking at. So without defining it for a student, I would ask you to look at this epithelial lining right here and tell me, is it simple or is it stratified? I'm not trying to trick you. It looks stratified, doesn't it? Okay, good. Okay, the second question then is it squamous, cuboidal, or columnar? Well, that cell right there looks columnar, but it doesn't seem to reach the apical surface. This cell looks flat. This cell looks cuboidal. You'll see that? So the anatomist looks at this and says to themselves, I don't know what to call this. It seems to be stratified, but it's not really squamous or cuboidal or columnar. What's worse is the tissue will transition. So if you look at it, when the bladder and the tubes, the bladder in particular, is full, it looks like stratified squamous. This is a picture when the bladder is empty. So, with no better way of describing it, it was decided that we would just call this a transitional tissue. Now, that's a cop-out. That's an anatomical cop-out on name. I don't know what to call it. It's a transitional. It is an important tissue, and we'll see it in the urinary system, lining the bladder and the ureters and the urethra. Transitional epithelium. Okay. We will begin on Wednesday with connective tissue and make our way through the connective tissues, and then we'll have a much shorter presentation on muscle and bone, and we'll finish that by Friday, I'm quite certain.